So as mentioned earlier, there were basically two studies in this one paper, a behavioral study in rats, as well as a genetic association human study. Here are results from the behavioral study in rats. On the left are the results from the object place recognition memory task, and on the right are the results from the Morris water maze task. Now remember, with the object place recognition task, if the rats explore the spatial displaced object more than the object that was not displaced, it is assumed that these rats remember the locations of the objects being presented to them. So in other words, if the black bars here are significantly taller than their white bar next to them, these rats formed and maintained a spatial memory. So where do we find a spatial memory? In other words, like where do we see a significant difference between the black bar, the height of the black bar and the height of the white bar? Well, when you stimulate this receptor with synktide, which is a neurokinin 3 receptor agonist. And all of these results really can be compared to rats given an empty vesicle. So that's what this VE represents here. But this effective memory enhancement in age rats after neurokinin 3 receptor stimulation was blocked by a neurokinin 3 receptor antagonist, which is what this SR stands for or represents here. So in other words, we no longer see a height difference between these two bars, whereas before we did see a significant height difference. This suggests that the memory enhancement we saw in the rats treated with synktide is neurokinin 3 receptor dependent. So here we have the Morris water maze. Now if you remember, in the Morris water maze, a declining mean time to find the platform with additional trials represents memory. And here you can see if we give synktide, so the white triangles, as you can see here, you get a sharp decline in the mean time to find the platform with additional trials. They start finding the platform much quicker each trial. Again, these memory enhancements are eliminated when you also give the rats a neurokinin 3 receptor agonist. So if we are now looking at these white rectangles or squares, you now see a lack of that memory enhancement that we saw prior. So in other words, in both behavioral experiments to test memory, if the rat was given neurokinin 3 receptor stimulation, in other words, synktide, they had better memory, and this enhanced memory is specific to neurokinin 3 receptor because we can block the memory improvements with neurokinin 3 receptor antagonists. So a good question to now ask is a neurokinin 3 receptor memory enhancement working the way the authors think? Is it working by increasing acetylcholine levels? Now this chart is really just showing the acetylcholine concentration in the hippocampus in the rats over time. If you just focus on the triangle line here, you can see, yes, the rats that had enhanced memory also had higher acetylcholine levels in the hippocampus. This is also true of the frontal cortex and the amygdala, all important areas for memory in the brain. So the authors have supported their hypothesis. But does any of this translate to humans? Obviously, we are ultimately interested in proving memory decline in humans, not rats. So as our method section stated, the authors looked at a population of elderly patients with mild cognitive impairment or dementia, found a series of SNPs in the gene that encodes neurokinin 3 receptor, and then asked, are any of the SNPs analyzed associated with Alzheimer's disease? And one SNP was found to have a link. RS2765. This may seem a bit confusing because the verbal memory task at the top here, a higher score means that they had worse verbal memory. But on the verbal memory task down here, a lower score actually indicates a worse memory. In both cases, dementia and mild cognitive impairment patients with the AA genotype, so shown here and here, of the SNP RS2765, had worse verbal memory than if they had the GG genotype, and those with one copy of the A performed intermediately well. We also saw a significant association between this SNP and hippocampal volume. If the dementia or mild cognitive impairment patient had the AA genotype, on average, they had a smaller hippocampal volume. So this genotype, AA, of this SNP, RS2765, is associated with worse verbal memory performance. It is in some Alzheimer's disease patients and is also associated with a smaller hippocampus.